Good day everyone. For today's class we shall be presenting a hibiscus flower. So this is the overview of the specimen. We shall be looking at the fundamental structures of this specimen. Looking at the overview, we have the pedicel, we have the receptacle, we have the epicalyx, we have the sepal, we have the petal, we have the long tube called the staminal tube bearing the filament and the anther. Then we have the stigma. So that's a long staminal tube. If you look at the whole thing, it has the male and the female part. So we look at the sepals, we do the dissection of the sepals and the epicalyx. Now we are actually going to remove the epicalyx, the first one, the second one, the third epicalyx, going to the fourth one, the fifth epicalyx, and we have moving straight to the last one, we have the sixth epicalyx. So if you look at the number of epicalyx and you can see the color, we have 2, 3, 4, 5, 6. There are 6 epicalyx and they are green in color. Looking at the sepals, you look at the pin down, we dissect the sepals too. We have a pin and a cut through the sepals. Now we have to remove all the, collectively they are called the calyx. Don't forget you have removed the epicalyx. We are removing the sepals now, which are collectively called the calyx. You can see their color. After you are dissecting it, after removing the calyx, you see the number of sepals we have. Now looking at it, we have a pin on the number of sepals. We have the first, second, third, fourth, fifth. There are 5 sepals and they are green in color. Distinctively we have 5 sepals. Collectively they are called the calyx. There are 5 in number, so the color is green. Now we'll move the petals. The dissection of the petals, we have a pin and a pin on it for the dissection, and we'll remove the petals one after the other. We have the first, we have the second one, the third, the fourth petal, and finally the fifth petal has been removed. Now what you have here is a long staminal tube which comprises of the male and the female part of the flower. Now you see where the ovary is. That is where the ovary is. You have the pedicel, the receptacle, where the ovary is being located, the long staminal tube, we have the anther and the filament, and finally we have the stigma. So what we need to do now is the dissection of a region of the ovary. We have the stigma. There are 5 stigmas. Then we have you can see the anther bears the pollen grains. So we have a dissection through the region, we are dissecting through. You will definitely see what it contains inside. Good. A cut through it, you see the structure we have inside. Now looking at the dissection of a hibiscus flower, we've separated every other structures, the sepals, the petals, the epicalyx. Now we want to separate the male part from the female part, that is the androecium from the gynoecium. The male part is the androecium, the female part is the gynoecium. So we'll look at the whole length of the pistil, which still bears the receptacle, the ovary, which contains the ovus, the style, and the stigma. So we are separating the male part now. The male part contains the filament, the anther. On the anther we have the pollen grains. So looking at the separated portion is the male part, then the whole length of that of this structure, whereby we cut part of the pedicel, is the pistil. And the pistil, you can see the 5 stigmas. The 5 stigmas, that's the whole length of the pistil, the female part, and we have the male part of the flower. So if you look at the other structures, we have, those are the petals, then we have the sepal, we have the epicalyx. So the dissection proper, we have seen different sections of the hibiscus flower. That's part of the pedicel. You have the receptacle, the whole length of the pistil. So we have seen different structures that makes up the hibiscus flower. That's the hibiscus flower before dissection. You see it before dissection. Then after the dissection, we can see the whole structures that makes up the whole specimen, which is the hibiscus flower. That's the hibiscus flower. You can see the other structures we have in it. Then we are actually done with the dissection. By now you should be able to state different parts: the petals, the sepals, the epicalyx, the pistil. Now we are going to show the dissection, the longitudinal section of the flower. That's the dissection of the longitudinal section. So when you look at the hibiscus flower and it is being dissected longitudinally, each half contains the same, exactly the same thing. This is the longitudinal section of the hibiscus flower. And when you say this, if it is dissected, it's cut along the ovary, so the ovules are evident, you can see it. So looking at this, we have the longitudinal section of the hibiscus